Om Shri Sai Ram. We'll start today's proceedings with a short prayer. Om Sai Ishwaraya Vidmahe Satya Devaya Dhimahi Tanna Sarva Prachodayat. Om Shanti Shanti Shanti. Jai Bolo Bhagwan Satya Baba Ji ki Jai. A small announcement before I hand over the stage to MSc brothers. The second speaker who was supposed to join virtually is in an emergency right now. So we will have the first talk. I requested Professor Jolly to extend his talk a bit more. So if we have any communication from the second speaker, we'll try to have the second session. Otherwise, we will end the first session little early. So then we can have a long refreshment break. Thank you, Sai Ram. Over to second MSc brothers to take the session forward.
With pranams at Swami's lotus feet, and my loving salams to each of the dignitaries, elders, guest speakers, volunteers, participants, and everyone physically and virtually present here. It is my privilege to introduce the first speaker of the session, Professor Mohit Kumar Jolly. Professor Jolly completed his BTech and MTech from IIT Kanpur in Biological Sciences and Bioengineering in 2010 and 2012 respectively. He earned his PhD in bioengineering from Rice University in 2016. In 2018, he joined IISc Bangalore as an assistant professor leading the Cancer Systems Biology Laboratory. His research focuses on developing mechanism-based mathematical models to decode cancer cell adaptation during metastasis and drug resistance in collaboration with experimental and clinical partners. He served as the co-chair of Mathematical Oncology Subgroup at the Society of Mathematical Oncology, and now serves as the editor-in-chief of NPJ Systems, Biology and Applications. Sir will be delivering his talk on the topic, Mathematical Modeling to Understand Cancer Cell Adaptation during Metastasis and Therapy Resistance. Please give a warm welcome to Professor Mohit.
First of all, Sai Ram, it's my great pleasure to be here. And I would like to thank the organizers, particularly Professor Vamsi, for giving me this opportunity to present here, to visit this magnificent place and to interact with all of you. I'll be sharing some of the work that we have been doing in trying to understand how mathematical models can be used to assess how cancer cells move from one organ to another, which is the process of metastasis, and how they develop resistance against different drugs that are given to cancer patients in the clinic. So I'll be presenting a mix of mathematical models, experimental data, and clinical data, and I'll be very happy to take questions at any part in the talk. So cancer, as many of you may already know, is a very dangerous disease, and in the past 50, 60 years or so, we have made some tremendous progress in reducing the cancer death rates, but for specific cancer types, not for every cancer type yet. But most of that advancements that have come because we are able to now detect cancer early and therefore start treatment early. It has not come because we have suddenly found a new cure for cancer. The other factors that have happened is, for instance, there is ample evidence now that smoking causes lung cancer. So there have been interventions programmed to reduce smoking and so on, which has reduced the rates of incidence and deaths due to specific cancers. But again, there are, in the clinic, there are two major challenges which we have not been able to get solved as a community. And what I'll present today is some of our work in that direction, working closely with clinicians and experimental colleagues to see if we can solve those two challenges. And those two challenges are listed right here. Metastasis, which is how cancer cells move from one organ to another, and therapy resistance, which is that various different drugs that we give to patients in the clinic, after a point, cancer just stops responding and keeps growing. The goal of those drugs is to reduce the growth of cancer, but it works for some time, but patients come back after many years, many months, depending on what cancer we are talking about, what stage the cancer is in. And at that time, there's just no therapy which can be given to control cancer at that point of time. So, sorry, next slide, please. Yeah. So metastasis, which is the major cause of all cancer-related deaths, basically refers to how cancer cells move from one organ to another. And this is one reason why cancer is a special disease. No other disease spreads to different organs in the body. Right? If you think about heart disease, it stays restricted to the heart. If you think of eye disease, it stays restricted to the eye. But cancer, irrespective of where it is born, which organ it initiates, it has the ability to spread to different parts of the body and grow cancers there as well. And which is what leads to multiple organ failure in cancer patients and eventually the death of cancer patients. So metastasis, this process is the reason for 90% of deaths of all cancer patients. And how this cancer spreads from one organ to another is largely through this freeway of blood vessels that we have in our entire body. So blood vessels connect the entire body. So imagine cancer cells in a given organ. They have access to these blood vessels which are running all throughout the body. So they can enter those blood vessels, travel to different parts of the body, and then when needed, they can take an exit from that blood vessel network or highway or freeway, whatever you want to call it, and then start growing tumors there as well. Now, one thing which actually caught my attention when I was starting to think about the process of metastasis is that it is a very inefficient process. So there have been studies showing that if you would place approximately, say, 10,000 cells in a primary tumor, which is where the original organ that cancer initiates, and these experiments were done in mice, of course, not in humans. So if you place those cells in a mice and then track how many of those cells were eventually able to develop tumors at a distant organ in the body, the answer was one out of 10,000. So it's very inefficient. 99.9% of the cells which leave the organ are not able to grow tumors. And despite this high inefficiency, it is still causing so many deaths. So you can imagine what would be the case if this process was more efficient, if there were more cells which were able to understand this. So what we are trying to understand is what is so special about that minuscule percentage of cells, 0.01% or 0.1% of cells, which are able to eventually metastasize. Because if we can target metastasis, there's no drug against metastasis as of today. But if we can target metastasis, then we can bring down the cancer death rates dramatically. So what do cancer cells need to successfully undergo metastasis? So think of the journey of a cancer cell. It leaves the home organ, it leaves its home, it travels in the blood vessel for approximately 1 to 2 weeks. It goes to different organs in the body, and then it has to adapt to the environment of that organ, which can be very, very different from the environment that it came from. So breast cancer cells usually go to brain or lung or liver. All of those organs have very different biophysical properties and biochemical properties as compared to what is seen in the breast organ. Similarly, prostate cancer cells usually go to bone, very different environment. So how are these cells able to, adapt to the environment around themselves that's constantly changing is the major reason here. So these challenges come in various forms. So the immune cells of the body are trying to kill the cancer cells. The drugs that are being given to patients in the clinic are also trying to kill the cancer cells. The environment around a cancer cell in terms of oxygen and nutrients available is constantly changing because it's not stuck at one location, but it's changing. It has to be, this is something I'll talk about in more detail in a minute. It has to be able to adhere to its neighbors, yet migrate at the same time. And it also has to be able to colonize, which means grow at the secondary organ. So what we need is to get a better understanding of how these different challenges come together and how is one cell, which is the successful metastatic cell, able to navigate through all these different bottlenecks and eventually grow a tumor at the secondary site. So the work that I'll be presenting is not just mine, is actually an integration of inputs that we have received from a diverse set of experts who actually treat patients, and of course academicians coming from different disciplines, different institutes and different departments as well. So I'd like to acknowledge them right up front, and I'll present a summary of the work we have been doing in the past 10 years to better understand this process. So if you think about the most common cancers that you might have heard about, breast cancer, prostate cancer, lung cancer, liver cancer, they all originate in what is called as epithelial organs in the body, which means that these cells do not migrate, do not invade. They are actually stuck in a specific three-dimensional geometry with very tight bonds with their neighbors. So how do these cells begin to move? By breaking the architecture that is present around them, is what is called as the process of transitioning from an epithelial state, which corresponds to these properties, to a mesenchymal state, which corresponds to the properties that these are invading, but they are not adhering. So if you think about red blood cells in the body, they are going all over the body. They are not stuck in a specific. So red blood cells are mesenchymal by nature. Most of the cancers I just talked about are epithelial in nature. So what cancer cells are able to do is that they leave these properties and they gain these other properties, which is what is called as epithelial to mesenchymal transition, or EMT in short. And that is usually the first step of them allowing to leave the primary tumor. Now, once they have reached the distant organ, they no longer need to migrate, they no longer need to invade, they actually need to settle down and grow a tumor there, which is the exact opposite of EMT, which is what is called as MET, mesenchymal to epithelial transition. So as you can see, this journey of a cancer cell involves reversible transitions between different states, each of which corresponds to a different set of property that is needed for the cell to survive at that point of time. Okay. Now, these are some schematics from research papers or review papers in the field of EMT, and which summarize what the role of EMT and MET is, which I already talked about briefly. And as you can see in all of these schematics, there are some assumptions hidden. And the assumptions are, A, EMT and MET is thought of as a very binary process, that cells are either completely epithelial or completely mesenchymal. That's an assumption. Another assumption is that a cell which will undergo E to M transition will necessarily come back from M to E transition as well. And the third inherent assumption here is that a cell has his own mind, and his or her own mind, and is making these decisions by themselves without necessarily getting signals from other cells around, right? So the decision of a cell seems to be independent from the decision of other cells. So these were the three assumptions we actually started questioning. That is EMT really a binary process? Is EMT really a reversible process? And is EMT really what is called in biology literature a cell autonomous process, which means that the cell is acting as an autonomous entity and making decisions by itself. Okay. So the approach that you will see today uses starting from experimental data, making mathematical models of underlying regulatory networks, making predictions from those mathematical models, going back to experimental data, and then seeing whether the predictions that we have been made are being validated, and if not, then how do you update the model constantly. So I'll just take you briefly into the literature of EMT to tell you what kind of experiments people do, what kind of data is present, and so on. So for instance, in this particular example, what you see are, these are two images of cell populations. Each structure that you see here is a cell. So here are epithelial cells grown in the lab. And then you treat those cells with a chemical which is known to induce EMT, called TGF-beta. And then you visualize the cells after usually 10 days or 2 weeks, and you see that the cell shape has changed, the cells are more elongated, and there are other experiments in this paper which show that these cells begin to move, these cells begin to invade, so on, right? So this is how you study EMT in the lab. Now, as you will very well notice that in this example, and therefore true for most EMT papers, they would take readings at only 2 time points, pre-induction and post-induction. And if you have only 2 time points, you can only say that a process is binary. Unless you collect data at multiple time points, you can't even reject the hypothesis that these are not necessarily binary, right? So again, there were many talks on COVID-19, for instance, at this meeting. So again, if I give you only 2 time points in this dynamic data and ask you whether there was a wave in between or not, there's no way you can predict it. You would need multiple time points to see whether there was a wave or not, right? So similarly, if you're given only 2 time points, how can you say that EMT is binary or not binary? You need multiple time points to be able to do that. So that data did not exist at that point of time. So we started by developing a mathematical model. What you see here are different molecules within a cell. They are all influencing each other, and I'll just show you the equations in a minute. So just as in Professor Banerjee's talk yesterday, he was talking about different cell types which are interacting with each other and influencing each other's behavior. Instead of cell types, these are different molecules within a cell which are influencing each other's behavior and controlling their concentration and time and so on. So what you see here is that these are 2 molecules in a cell, which when the levels of these molecules is high, the cell is usually mesenchymal. But if the levels of these 2 molecules is high, the cell is usually epithelial. Okay. Now, each arrow that you see here is some sort of quantitative input-output relationship, and I'll show you the examples in a minute. And these kinds of models have been extensively built for bacteria, for yeast, and simpler organisms. So when we started working on this around 10 years ago, we asked the question, can we now develop these models for cancer and see how cancer cells behave and see. So now this is the model. Again, I'll not go too much into the biology. Let's look at the equations. There are 6 equations, 6 molecules which are all influencing each other in different ways, shown by this set of coupled ODEs. Each of the molecules has a rate of production that is highlighted here. Each of them has a rate of degradation based on their half-life. Each of them is being influenced by other sets of molecules. So those are the terms that you see here. And these are typical functions, saturating functions, either of increasing or decreasing monotonic nature that you see here. So what did the model predict? Here, x-axis is some sort of EMT inducing signal. Y-axis is the measure of EMT-ness in some sense. And the solid blue lines, as you can see, are stable states, and the dotted red lines are unstable states. So what you see here is that this system can be tri-stable in nature. You can have 3 distinct equilibrium points which can all coexist at the same parameter region. Okay. So if you have very low levels of this molecule, Zeb, which is shown here, the cells are epithelial. If you have very high levels of this molecule, the cells are mesenchymal. But in between epithelial and mesenchymal, there is a 3rd state which is possible that was not really captured in the literature earlier. So this is what we called a hybrid epithelial mesenchymal phenotype. These cells can have the property of both migrating and invading at the same time. So these cells are not moving as single cells, but they're moving as a group, as a unit, as a cohort, as a cluster, and they're able to metastasize into that mode as well. Okay. Then you can also see there are regions where there is monostability. There's only one range of one stable steady state available, to another steady state available, the other extreme mesenchymal, and in between you have these regions in which there are 2 states or 3 states which are available. Okay. So these were the predictions that our model made, that EMT is not binary. You can have cells in a hybrid epithelial mesenchymal state. The second prediction was that now if you take the cells with the same background, genetic background, you can still have multiple states, which is what you see in the bifurcation diagram, and that the cells can also switch back and forth without a whole lot of induction. Okay. So these were all the predictions. Then we started working with experimental colleagues to test these predictions, because at that point still EMT was thought of as a binary process. So what you see here is images of lung cancer cells grown in the lab, and they have been stained for uh, two different molecules, red and shown in red and green, which one represents epithelial, one represents mesenchymal. So what you see here is that individual cells have both red and green molecules, saying that these are actually hybrid cells, which can maintain themselves in the lab for multiple durations of 2 months. So that's the first predictions, that this is a stable state that you can see. Then you can quantify this further. You can measure the levels of each of these molecules in breast cancer, prostate cancer, lung cancer, colorectal cancer, all different cancers, and you again see that cells belong to multiple different states. They lie in all different quadrants. So this is the mesenchymal quadrant, this is the epithelial quadrant, and this first quadrant is where the double positive or the hybrid quadrant. So those cells are present there. And now if you segregate these cells in the lab, there are methods to do that, then you see that they actually switch to the other cell type. So even when there is no induction being given, these cells can actually switch back and forth in the lab. Now, these predictions that there are hybrid states or the EMT is not a binary system, in the past decade, there have been many models, mathematical models that have been made, each corresponding to a different network or each corresponding to a slightly different modeling style. And some have come up with saying that there is one state, some say there are 2 states, 3 states, and so on and so forth. But clearly, EMT is not a binary process. That is something that has been very well established in the literature by now. So these are just different examples of mathematical models that people have made, showing that on this epithelial and mesenchymal, if you look at this 2-dimensional plane, there are stable states which are on either extreme, epithelial high, mesenchymal low, or mesenchymal high, epithelial low, but in between also you have many hybrid states that depend on which cancer you're looking at and which molecules you're looking at. Okay. So that's fine, they exist. EMT is not binary, that is well established now, but why does it matter? What is the role, if any, of these hybrid cells in the process of metastasis? So that was the second question we started asking. Now, if you think about the properties that a cancer cell would need to be able to grow a tumor at the other site, those properties are very similar to what are called as stem cells in our body. So stem cells are those cells in our body which are capable of giving rise to multiple cell types. So our life starts with a single cell, right? And that single cell then gives rise to 10 to the power 14 cells eventually into a fully functional human per se. So that initial cell is what is, and many other cell types are what are called as stem cells, saying basically the terminology coming from that everyone else stems from them. Okay? So cancer cells also need to have these properties of stem cells so that they can grow a tumor. When a single cell reaches a distant organ, it has to be able to give rise to multiple cell types that will constitute a tumor at that particular organ. So we made a mathematical model for that in terms of what molecules control EMT, what molecules control stemness, how are they connected to each other, can you write them down as a set of differential equations. So what our model predicted was that we had a very similar set of equations that you saw earlier. We identified that these hybrid cells are actually having the highest stemness. So the epithelial and mesenchymal, they are capable of giving rise to tumors, but not the maximum. The maximum properties are actually present somewhere in between. So that is why they matter. So this was just a prediction at that point of time, but now over many years, there have been many papers showing that these hybrid cells are actually most metastatic. So here in this example of skin cancer, you see that they identified experimentally 6 different states of EMT, and then they compared their ability to metastasize in mice and showed that the highest ones were not the completely epithelial or completely mesenchymal, but a subset of population which was present somewhere in between. There was another paper which actually showed that in breast cancer, this is skin cancer, but in breast cancer also they showed that these hybrid cells are actually essential to form tumors. So the experiments they did was they took some epithelial cells, put them in mice, see how many tumors grew. They took some mesenchymal cells, repeated the experiment. They also took a mixture of epithelial and mesenchymal cells, repeated the experiment, and then they took only hybrid cells. So hybrid cells were the ones which were actually forming most metastatic, of the order of 10 to 20 times more metastasis than epithelial, mesenchymal, or mixture of them as well. So this further supports the predictions that we made before any of these experimental data came out, was that these hybrid cells are actually the most metastatic one, are the most dangerous ones. So that's all still looking at mice data, experimental data. Let's go to patient data and see whether our predictions are supported. So here what has been done is data from breast cancer patients have been taken. Each dot that you see here is a patient. The y-axis is the number of these hybrid cells, and the x-axis is 4 different classes of breast cancer, starting from the least aggressive to the most aggressive. And again, what you see is that the most aggressive cancers have the highest number of such hybrid cells. There was further analysis which was done in many different cancers. These are just examples. So the question these authors were interested in asking is, what is the threshold percentage of hybrid cells that a patient needs to have such that his or her survival in clinical data is much worse? Okay? Do you need a large percentage of them, 20%, 50%? What is that percentage? Their answer is 2%. So if a patient has only 2% of these hybrid cells, that is sufficient to give a much worse survival. So here the x-axis is time and the y-axis is probability of survival. And overall survival means that when this probability is not 1, means that patient is dead. This is disease-free survival, which means when the event is false, then cancer has come back. So this is life and death, and this is cancer present or cancer absent in that patient. Okay? So again, you can see that even a small percentage of hybrid cells are actually able to make the cancer much more aggressive, leading to a much faster death of patients or much faster recurrence of cancer as well. So in the past 10 years, now the field of EMT has adapted itself, and there is more and more literature showing that EMT is not binary. And there are one or more states which has been seen in mice, which has been seen in patients. And these hybrid cells are the ones which are actually maximally metastatic as compared to the completely epithelial or completely mesenchymal ones. So then we started looking at this data more carefully, is that why are hybrid cells most metastatic? So far we have said that yes, they exist. They have high stemness, so therefore they can form tumors. But is that it? Or are there other reasons as well which make hybrid cells so much more aggressive? So this was the clinical data. So in this schematic, what you see is that they compared the ability to proliferate, which means to divide, to invade, stemness, metastasis, and this is the property I'll talk to you in a minute, something called plasticity. Plasticity means adaptability. That whether a cancer cell is able to change its state and go to another state if there is stress around it, okay? Because that is what will allow it to survive for a long period of time. If it doesn't adapt, it will die eventually, okay? So what they found was that this plasticity was also actually highest in these hybrid states as compared to the epithelial or the mesenchymal state. So we started asking the question, why are hybrid cells most plastic? Is there some inherent property of these networks that we have been looking at? So this work was led by my PhD student Kishore, and started asking, if you look at the regulatory networks, can that explain why do you see high plasticity in this? So now in the past decade, the models of EMT have been growing in size. There are more and more molecules which people have now identified and how they interact with each other. So instead of taking an ODE approach for such large systems, we took a Boolean modeling approach, which basically means that a gene is either on or off. You just have 2 states, on or off. And the modeling strategy is very simple. What you do is, because the state is defined as just +1 or -1, you count in for a given node in a network, how many different other nodes influence it, and then take a weighted sum of them and call it 1 or -1. It's a very simple strategy. Okay. Now, what you see here is in this one of the large networks that we looked at, if you represent this network as a matrix, which basically means that each row is the source, each column is the target. Wherever there is an edge present, the edge can either increase its levels or decrease its levels, so it's either activatory or inhibitory in nature. So that is what is shown in red and blue respectively. So in this 22 cross 22 matrix, only 82 entries are filled and the remaining 402 entries are empty. That's why it's a very sparse matrix. Now, we very well know that both in biology and in life, the influence does not need to be direct. You meet someone who meets someone else, who meets someone else, and the influence can pass on, right? So similarly in biological networks, if node A influences node B, which influences C, which influences D, the effect of A on D can be both direct or indirect, depending on how many intermediaries are present in between. So then what do you do to capture that indirect effect is just you multiply this matrix to itself. When you multiply this matrix many, many times, you see a very clear pattern that shows up. And the pattern is the following, that there are two sets of nodes. One set of nodes, so if you're talking about the interactions within that set of nodes, they're all increasing each other. They're all supporting each other, activating each other, which is what these two boxes are. But if you look at the influence going from one set of nodes to the other set of nodes, they're all inhibitory in nature. So it's like two teams of players which are playing a tug of war. So within a team, there's a lot of support. Everyone is supporting each other. And across teams, team 1 is trying to make sure that the molecules of team 2 go down, and team 2 is trying to make sure that the molecules of team 1 go down, right? So it's a constant struggle that is going on between them. Now what are these two teams? These two teams are players that support epithelial and the players that support mesenchymal. There is no team for the poor hybrid cells. They're stuck in between. So again, imagine a tug of war kind of a situation. If you keep something, right at the rope, somewhere at the center, that will have the maximum tendency of falling off when one of the team is pulling or the other team is pulling. That's what is happening to hybrid cells. Hybrid cells are somewhere in between. They have no support system. They have no mutual activation going on between them, and that is the reason why they are most plastic. The epithelial cells, it takes a lot of external stress for an epithelial cell to leave its behavior. It takes a lot of external stress for a mesenchymal cell to leave its behavior. But hybrid cells they're not committed to anyone. They can easily switch. And we showed this via detailed simulations. We actually showed that we asked the question as to what is the level of perturbation you need for an epithelial cell, mesenchymal cell, and a hybrid cell to leave their behavior, to go to a different state, because there are multiple states available. It's a multi-stable system. So what we saw was that if you're in an epithelial or a mesenchymal state, you need to give the perturbation which is almost 7 to 10 times more than the perturbation that you need to give to a hybrid state to leave it. Okay. So again, those properties, we all sort of quantified this. And it's the absence of any teams of these hybrid cells that actually make them the fittest for metastasis. So in this first part where we were asking the question, is EMT binary? The answer is clearly no, it's not binary. And the ones which are hybrid cells are actually the most metastatic. We have other work which I'm not showing here for lack of time. They also are able to resist chemotherapy and targeted therapy, and they end up being most plastic and so on. So this was an example where mathematical models drove new predictions, which were then tested in the experimental data, and then also in the clinical data. There was more and more evidence that has come up. The second question is also very simple: is EMT reversible? So the idea in the field had been that these cells, once they leave the primary tumor, they switch on EMT, and once they reach the other organ, they switch off EMT and go back to being epithelial and start growing tumors there. Now, there has been some literature which suggests that that might not be as simple, because when they did experiments in the lab similar to the ones I showed you earlier, what you see is that if you go take these cells in the lab and treat them with EMT inducing agents such as TGF-beta, for different durations. You treat them for 2 days, for 5 days, for 8 days. And then you remove the signal, you remove the chemical, and see whether cells go back to being what they were, right? So if you remove the signal after 2 days or 5 days, they go back to being epithelial. But if you remove after 8 days, they do not. So there is some tipping point behavior that is being seen here, which makes the process either reversible or irreversible. So we wanted to ask, what is the tipping point? How is it characterized at a mechanistic level, and how can we better understand it? So we made a very simple phenomenological model here. What we said is that we all again know that the influence, again in taking examples from life, the influence of a person doesn't necessarily need to disappear when that person is no more around us, right? Some people can make such dramatic influence on our life that it can continue even when they are not physically around us or very close to each other and so on. That the same thing can happen in biological networks. So a gene can be turned on, and even when it has turned off, it might have made other changes in the biochemical reactions that are ongoing, such that its presence or its impact or its influence can continue to be there even when it has turned off. Okay? So it's a long-lasting memory kind of behavior that can be seen. So we recapitulated this using a very simple mathematical model where one of the terms which was a constant in the previous set of equations that I showed you also ends up becoming a dynamic variable, and that can continue to decrease as a function of time depending on how long a function was on. So the duration for which a gene is on, if it was on for a very short period of time, it has very less memory after that, but if it has been on for a long period of time, then the memory component grows and it stays there and so on. So what we saw was that when we do this in the network that I showed you, it can actually change the population distribution dramatically. So if you include this memory component in this one interaction, this is the population distribution of stochastic simulations before you incorporate these terms. So what you see is that in this population, around 80% cells are epithelial and 20% are mesenchymal or hybrid. But if you incorporate that, the proportion of mesenchymal increases dramatically and the proportion of epithelial decreases dramatically, right? So this can actually change the population structure quite a bit. So then we collaborated with an experimental colleague who did these experiments, and in a much more careful manner than what were done earlier, asking the question, is long-term EMT always irreversible? Okay. So now the experiment that was done was the following. You induce with TGF-beta for 13 days and you wait for 18 days. But when you induce for longer durations such as 22 days, don't wait for 18 days, wait for a much longer period of time, 45 days, because earlier data just ended here and it claimed that EMT was irreversible. But what if it was reversible at a later stage of time for which there was no data that we collected at that point of time? And we exactly saw this. So this is the green one shows the long-term duration. So x-axis is, so now this is the withdrawal dynamics. The x-axis is how long the signal has been gone, and the y-axis is where it started from before induction. So the induction was able to bring this signal almost close to zero, but almost for 20 days it stays zero. And only after 20 days it actually starts improving and increasing the levels, right? So earlier experiments ended here, and therefore claiming that EMT was irreversible. But now if you collect the data later, you actually see that the cells come back and they become epithelial again. It's just that they take a longer time to come back, okay? And this we saw, you can measure different molecules, and we saw the same trend. There was some delay, but that delay eventually led to where it had to lead to and so on. So there is memory that is built in the system, yes, but that memory can also decay, right? And how does a memory decay? How again, taking examples from life, memory usually decays at time scale of generations, right? When there is a next generation of people coming in, the transmission of information of memory from one generation to another is usually not as strong. And then if you look over many generations, that memory can decay. So what is the equivalent of generation in biology? Cell division. Every time a cell divides, it gives rise to 2 new daughter cells, and so on and so forth. There are errors in this process, and those errors can keep accumulating and remove the memory in the system, which is what we saw. when we do these simulations for longer durations, which correspond to approximately 10 cell divisions, which means 10 generations, again going back to the analogy of life, you see that the memory has disappeared and the cells continue to behave as what they were even before. So what we are saying is that yes, there is memory, but that memory is finite. It's not infinite. And the time scale of deletion of that memory is much, much longer as compared to the time scale of these biochemical reactions that are happening. So cells can be resistant to EMT. So they can be stuck in an epithelial state, they can be stuck in a mesenchymal state, and they refuse to undergo these transitions to varying degrees, and that is what controls the time scale at which these populations revert or don't revert. The third part is EMT cell autonomous? Is the decision whether a cell undergoes EMT or not taken by an individual cell, or is it decided by its neighbors as well? Is there some consulting going on at a spatial sort of community level as well? So now there is this experimental data which shows that when cancer cells leave the primary tumor as a cluster as compared to leaving as a single cell, they actually form much more metastasis than what they did when leaving as single cells. And these clusters, so here the figure is 50 times more, and these clusters are not 50 cells large. These clusters are usually 7 to 10 cells large, but they have a disproportionately high advantage in forming metastasis at that location. Okay? So we wanted to ask, what leads to the formation of these clusters? What decides whether a cancer cell will leave as a single cell, or a cancer cell will leave as a member of a cohort of cells or a group of cells which are eventually able to form many more tumors. And again, there has been data showing that these clusters are formed before they enter the circulation. So it's not that in circulation or in bloodstream, two cells come together and form a cluster and then it's formed before. So it seems to be an intelligent decision making on the part of cancer that I'm going to send some single cells and I'm going to send some clusters with the hope that, because what cancer is trying to do is survive. Cancer cells are our own cells in the body which have started misbehaving for whatever reason. So just as when there's an injury in the body, our cells try to recover from that injury. Similarly, when cancer cells are facing stress, they're facing lack of oxygen, they're facing lack of nutrients in the environment around them, then they leave to other organs to form tumors there. Because the major property of cancer cells is that they divide much faster than our regular cells in the body. And when you divide much faster, when you grow much faster, you're going to face a limitation of nutrients, you're going to face a limitation of resources, and that will trigger the movement to different locations to look for better opportunities there. So, so that was the question we started with, is that how do cancer cells form these clusters before entering the circulation? Because if they're doing it before entering the circulation, which means that the decisions of EMT or MET are not dependent on individual cells, they are decided together, collectively, at a population level before they leave the tumor itself. So now the question is, how are clusters of circulating tumor cells formed, right? So again, try to draw an analogy. In a room full of many people, if there are two hybrid cells which want to form a cluster, because the epithelial cells cannot form a cluster, mesenchymal cells cannot form a cluster by themselves. Epithelial cells, they don't have the property to move. Mesenchymal cells don't have the property to adhere. So hybrid cells have to bring everyone together to form a cluster and then possibly move. So if there are multiple ways this can happen. So let's say there's a hybrid cell here and a hybrid cell here. How do they talk to each other? How do they send signals to each other? And if they are able to send the signal and the other hybrid cell is able to receive the signal, and how does that hybrid cell then physically move closer to the other hybrid cell to form a tumor? So that's one option, that there are two hybrid cells somewhere, or two or more hybrid cells somewhere in the sea of cells, and they communicate, and one of them moves or they meet at a common location. This person is coming from here, this person is coming from here, let's meet midway, form a cluster and leave. That's one possibility. The other possibility is that there's a hybrid cell which is able to change its neighbors, which is able to convince its neighbors, why don't you also become hybrid and then we'll go together. That's another possibility. So in that, there's no movement involved. There's just convincing involved of your neighbors, and it's probably an easier process to do. So these were the two hypotheses we looked at. I mean, this one, we did not find any extensive biological data yet. So we focused on this one, asking that can cells influence their neighbors and coordinate their behavior and live simultaneously. So how do we go from intracellular models to population level models? Like, this is what's going on within a cell, and this is a very different time scale, length scale, and so on and so forth. So one of the ideas we had was that we all know that in various different scenarios, and again going back to Professor Banerjee's talk yesterday, these cells talk to each other. They exchange signals, they exchange nutrients, they exchange various different things, right? So it's possible that these cell-cell signaling might be able to give rise to these clusters. So there's one set of cell-cell signaling, and I'm not going into too much biology details, but what you see here is this is the same circuit that we saw earlier, and this is the cell-cell communication circuit. They're all cross-talking to each other, so the variables are interlinked. The coupled ODEs will have each of the other forms. And this is a cell in the boundary here, and this is another cell. So you can see that these molecules are also talking to cells, the other cell, and this is going to influence EMT in the neighboring cell, and so on and so forth, right? So what we then did, we took a grid of cells. So this is 50 cross 50 cells. So each cell has 10 ODEs in it. So this is 25,000 coupled ODEs that we are solving simultaneously, and asking the question, is there a pattern of behaviors that you see? So now these are two, again, think of them as two different parameters in the model. And what we saw was that when the value of this parameter was high, you could actually see hybrid cells which are right next to each other, and they seem to be forming a cluster. But when the value of this parameter is high, that feature was not seen. So what we are saying is that this particular molecule is contributing or may be contributing to formation of clusters of hybrid cells. Now this is just a prediction. Let's go back to experimental data. When you compare the cells moving simultaneously and cells moving as individual cells, you actually see that the levels of this molecule is higher in the cells that you actually saw as moving together. And these cells are also hybrid in nature because they are lying in this first quadrant where epithelial and mesenchymal markers are both high. This can also influence spatial patterns in a tumor, which have been seen experimentally, that there are more mesenchymal cells at the edge and more hybrid cells at the interior. So we further did these experiments. I don't have the data here. We did these experiments where we actually deleted these molecules from the cell and showed that metastasis went down dramatically. Okay, so which means that this molecule is playing a role in forming clusters and allowing cells to move, which is what our model had predicted. So cell-cell communication, it can be via immediate contact, or it can be also via one cell is secreting something which is being sensed by the other neighbor. Both of them can impact the spatiotemporal patterns of EMT within a tumor, and therefore change the rates of metastasis and so on. So I'd like to summarize with three different questions that we started with. And in each of them, you would see instances of models driving new hypotheses. Is EMT a binary process? And the answer is clearly no. Is EMT a reversible process? And the answer is not always. It depends on how long have you induced. It depends on how long have you withdrawn the signal. And is EMT always autonomous? The answer is again not always. It depends on what is going on within the environment of a cell. So again, this quote, many of you may have already heard of. Essentially, all models are wrong, but some are useful. So I'm not saying that this model, of course, solves every question out there in metastasis, but this is a way of thinking about it. And this, when we also have all these experimental models of cancer. So what these experimental models mean as I briefly alluded to during the talk as well. So what people would do in the labs is try to treat cancer in mice, right? So, or try to induce cancer in mice. Now, why mice? Mice is not the perfect model system to study human behavior in any sense, right? But you can't do these experiments on humans. And PhD students need to graduate on time, so you need to choose an animal which has a smaller lifespan, which can be either killed by the tumor or die of itself before the time scale of submission of a PhD thesis, which is usually mice don't live that long, right? So all these practical constraints, we choose certain models. So similarly, mathematical models, you might say that we have not included 5 other different things which are going on in the tumor. Definitely, yes, and the models can be improved and the models can be more encompassive and so on. But here, again, the goal is, can we drive new hypotheses, or can we expose assumptions? So for instance, the assumption in the EMT field was that the stemness in mesenchymal is higher than stemness in epithelial. Without collecting any data of hybrid, they just made a linear hypothesis. But only when this data was collected, now we know that it's a non-monotonic behavior, and the ones which are actually most metastatic are the ones which are somewhere in between. Another assumption which was there in the field of EMT was that time to revert to epithelial increases dramatically depending on how long it. So this is what the assumption was, that the process is irreversible, but we showed that it's not irreversible. If you actually wait long enough, cells do come back, right? So these assumptions or these which are corresponding to our linear or binary thinking were shown to be false by the mathematical models, which then drove the new hypothesis here. So clearly cancer is a complex, dynamic and adaptive system. There are many, these are our own body cells which are trying to adapt, and we cannot think of cancer as a static system which is just sitting there waiting for us to give the drug and die off. So we need to think about very carefully about which drugs or which therapies are going to induce which kind of side effects. So for instance, in the early 2000s, there were clinical trials being done on these drugs where the idea was that let's block the blood vessel growth of cancer and we have solved cancer, because if you stop the blood vessel growth, you deprive them of food, you deprive them of oxygen, you deprive them of nutrients, and you will kill the cancer cells. That was not the case. Patients who actually received these drugs started dying faster than patients who were not receiving this drug. And now we know, 20 years later, one major reason why this was happening was because when you were giving that drug to patients, you were increasing metastasis. Because these cancer cells, you're depriving them of oxygen and nutrients at that location, but then you're making them much more aggressive because now they are in search of new opportunities at different organs, and they just, so it's a major side effect, right? And it's you might think it's non-intuitive, but if you think of cancer as a static system, if you think of cancer as a dynamic system, it's not non-intuitive. That's what someone would do if they have to survive in different scenarios. So that's why I think mathematical models can play a crucial role in understanding how cancer cells move from one organ to another, and also understanding how to give therapies, which therapies to give. I mean, the therapies that are given to patients today in the clinic, many of them have not changed dramatically in the past 50 years. Or 30 years or 40 years, depending on which cancer we are talking about, right? We can't think of a word going back 30, 40 years ago in terms of other technology, right? This technology did not exist dramatically in 40 years before. So why have we not made as much progress in better targeting cancer? One of the reasons I think is because mathematical modeling approaches have not been integrated. It's still largely empirical studies that have been going on. And that is where my request to all of the students here is to start thinking about various different diseases where also you can apply whatever different terminology, techniques and tools that you're learning. And I'd like to thank Professor Vamsi again for putting together such an excellent conference where you're all exposed to these ideas, and I'll be happy to chat further. So with that, I would like to thank the fantastic set of people who are part of the group who come from different backgrounds, maths, bio, physics, chemistry, engineering, cancer biology, etc. They all work with each other, they all teach each other, and try to solve these questions. With that, thank you once again for your attention. I'll be very happy to take questions.
Thank you, sir, for the wonderful talk. Before I open the questions for the house, I have one quick question, sir. So, do you also work on the data aspects, sir? I mean, machine learning techniques, you collect data of the cancer, because we also just recently started in this field of metastasis.
Right.
So, would like to know any machine learning or deep learning techniques what you have used.
No, that's a very good question. I think with the extensive data being collected, those technologies can, or those techniques can be extremely helpful. We have used it only in one project so far, where we were trying to predict which cell is epithelial, mesenchymal, or hybrid based on molecular characteristic. But that's about it. We have not done it further, but I'll be very happy to discuss and see there are ways to work together on that.
We look forward for that, sir. Thank you. So now I open the question and answer session for the house. Anyone from the speakers?
Sir, thank you so much for making us to know about the many things related to the epithelial cell and mesenchymal cell and the hybrid cell. So actually I am curious to know that because there was some research done, Nobel Prize research, that fasting also is responsible for killing of the cancer cell. So now I am knowing the new things that when the cancer cells are not getting oxygen and food, then they are going to be, they are shifting the organs, right, to other cells. They are going and they are generating, they are going to generate the metastasis. So just I am looking that how these two things are looking different, right, contradictory to each other. So can you enlighten us about this?
No, that's an excellent question. Thank you. There is literature, as rightly pointed out, that fasting, and especially intermittent fasting, or also not having high-fat diet, for instance, can play an important role in controlling cancer. I'm not sure if it is about metastasis, first of all. It's more about cancer initiation, from what I know. And secondly, fasting is not depriving of oxygen for a very long time. So fasting is depriving it maybe for the order of a day or something, or a few hours. But therapies are so it's the difference between an acute stress and a chronic stress. In one case, you are giving, it's like a pulse. So you suddenly deprive them of oxygen, but then after a few hours, you let them have oxygen. Then you again deprive them of oxygen, let them have oxygen. So it's a very oscillatory kind of a system. In the other one, you're suddenly shutting down oxygen and then just not letting recuperate. So those are very different stresses, and cells may adapt very differently to that. So in fasting, they might just wait around for, because cells divide usually every 24 hours. So if within 24 hours they are getting oxygen and nutrients again, they may not have as much of an incentive to go to other organ. But in the therapies, the stress is much longer. So that's what I currently can comment on.
Thank you so much, sir.
Papan, sir, any question? Any questions from the part?
Thank you. Thank you for so nice talk. I have a couple of questions. So in the third part of your talk, so you're telling about two models. Like, did you use a multi-scale model there?
Yeah, so these were multi-scale. They were multi-scale inherently because you're looking at both what is going on within a cell and what's going on at a population level.
In your theoretical work, what you presented in the third part,
Right.
so how could you manage the different time scales?
So no, the time scales here are only, it's a static lattice. There's no cell division, cell growth, cell birth happening. There's only biochemical reactions. So there's only one time scale here. There are not, so it's not multi-scale in the true sense, where there is all these other processes happening. It's multi-scale in a sort of pseudo sense, because there are multiple scales involved here in terms of length. And
For the first model, where you have on the molecules of the compartment
Yes.
of the cell,
yes,
that has one different time scale.
right.
and later when these clusters will have a different time scale.
Right, sure. So, but we are not looking at migration because we did not go to the hypothesis that these cells migrate and form a cluster. We stuck to the hypothesis that these cells influence their neighbors and that neighbors' influence can be captured through the similar set of molecules.
And in the first model, where we have the molecules in a cancer cell,
Right.
how could you get those parameter values? Is it through blood test or is it through the cancer cell?
It's mostly from
Helping-
experimental literature. So there is literature where you would, no, it's not from patient directly, it is from experimental literature where you would take these cells in the lab, grow them and do these experiments where you, let's say, increase the levels of a molecule and then see what is the effect on the other molecule. So those kinds of experiments have been done in ample amounts where you can get this information.
And this hybrid cells concept, is it there in humans also, or it is only proved in?
Excellent question. Yes, we have hybrid cells in our body in many organs, and what is their function, we still don't completely understand. The hypothesis in the field is that these are the ones which are responsible for quick response to stress. So liver, for example, which is has the highest regenerative potential organ in the body. When liver damage happens, then these hybrid cells are quickly responding to the damage and able to control the damage. So it has a very homeostatic role or a physiologically important role. Somehow cancer cells seem to have hijacked that and use it for their fitness as well.
And one question out of curiosity, like post-COVID, there is so much importance on health. People are giving so much importance, and these things like full body checkups and blood tests is becoming common. So in that blood test, can we able to tell whether some hybrid cells are moving, and can we detect early cancer? Is it helpful?
Absolutely, yes. So blood tests are becoming, liquid biopsy is what it is called in the clinic. It's becoming more and more prevalent that you can look at blood and identify whether there are cancer cells or not. The technology is still developing, I would say, because for every 10,000 red blood cells, you have one cancer cell. So the ratio is also so skewed that identifying that one cancer cells in that technology is still being developed.
Thank you, sir. Thank you.
Any questions from the participants? Yeah, finally.
Hello, sir. So I have one question is, there are companies like DeepMind and Babylon who are working with the data-centric approach, like say for example the protein folding problem, and they're trying to understand the protein level version of it using deep learning. And on the other side, there is a cell level research which is approaching with the mathematical aspect of it, which doesn't involve the deep learning. So as you being one of the most important persons in these field, which would you suggest is more effective? Is the deep learning version of dealing with data, or is it the theoretical mathematical version of dealing with math and the cells?
No, that's a question I ask myself every day. Thank you for bringing that out. And I don't think we should pit them against each other. I think there is a way to integrate them both. And you're absolutely right that in molecular level details such as protein folding, deep learning has come a long way. One major reason for that is because we already have structures for many. Learning basically means you have a lot of data to train on, based on which then you can make more accurate predictions. So protein folding, the laws of protein folding are reasonably established. It's energy minimization principles. There are enough protein structures deposited in protein data bank and so on, based on which you can train. Here, on images, there might be enough data, which there can be training. I'm not sure if at a cellular level there is enough data. If that is being collected, definitely there are ways to, there is definitely a huge value in using deep learning approaches also here. But I think that benchmarking is not yet reached that stage in cell biology, which it has reached in molecular sort of protein folding kind of constant. So, but having said that, as and when it reaches there, definitely, hopefully in next few months or years, there's definitely value in integrating both of them. I don't know enough about the protein folding literature to comment on that. Yeah.
Any questions from the virtual participants? Since already we have taken in quite a number of questions. So I'm sure all of you have enjoyed Dr. Jolly's talk. So let us give a big round of applause. I have a couple of announcements before we break for the refreshments. So we'll all now we'll take a half an hour break and we'll all reassemble at 11 o'clock. So we have an interesting talk from the actual team of Department of Mathematics and Computer Science. Then we have the final valedictory session from 11:30 to 12 and the conference ends then. Okay, thank you. We'll break now for the refreshments. Om Shri Sai Ram. Welcome back to the last session, last technical session of the conference. Over to Second MSc brothers to take the session forward.
Sairam and good morning to all. It's my humble duty to introduce the next speaker and also the last speaker of 3 days National Conference in Mathematical Biology Theory, Methods and Applications held at Sri Sathya Sai Institute of Higher Learning, Prasanthi Nilayam campus, Mr. Sathya Sai Mudivanda sir. Sir has 32 years of senior management experience in actuarial and technology, working in 9 countries across the globe. They have successfully completed 36 large international projects worth 25 million USD with teams of up to 500 members. Sathya Sai sirs holds 5 professional qualifications in actuarial science, insurance and management and has published 36 research papers in international journals. His team has also authored 7 articles in actuarial magazines and presented at conferences, with 27 dissertations guided and co-guided at the postgraduate level, 12 years of teaching experience, and mentoring 3 scholars to receive their PhDs. Sathya Sai sir's contributions have been recognized with 3 organization level awards, including the CAS Award, University Award 2022. Now please join me in welcoming Mr. Sathya Sai Mudivanda sir. Sairam.
Om Sri Sai Ram. Invoking divine presence here, I welcome all the dignitaries here, participants, speakers, our colleagues and dear students. So this is a great opportunity for me to bring up the actuarial science, how actually we work with biologists in practice. So this is going to be a little what is actuarial science at the beginning, few minutes of it. Then we'll have what is the life perspective, and then we jump into the concepts of actuarial science that we use in biology. Then we have a model which actually very recently that got published in 2019, which we are using it for our purpose here. So that is the agenda we have. Can you please move on to the next slide? So joining me, along with me, Dr. Rohan Yashraj Gupta. He is online, joining us. So he is the first PhD in India in actuarial science. It's produced from the same, our own institution, 2 years back. And he currently works with Accenture in a senior management position. He published a lot of papers in actuarial data science area. So he will actually bring up a lot of perspectives on the actuarial data science. He presented to multiple audience in North America, Asia, and India. So recent one was Global Conference of Actuaries in New Delhi. So please move on. So agenda is this. So we move on, that's what I discussed. So actuarial science primarily deals around 3 words: life, death, and money. So life, when we say life, actually it includes everything, where we can look at actually the longevity, survival, and other things. Death is actually when it ends, the life ends, that is a point actually we are looking at. Then third one is, for both the things, you need money, okay? So money, when we are talking about the money wherever it moves. It moves in Wall Street, it moves in Bombay Stock Exchange, it moves in commodities, it moves in every other area. So fundamentally, so what is actual science? That actually we can go deeper, but these are the three keywords which we actually operate in our practice. So in the current world, the issue, major issue is longevity. People surviving for a longer time is of more concern than actually the death. Because death, there are ways actually we can avoid it by improved medical situations, but like at the end of it, it impacts the people when they survive for a longer period. When they survive for a longer period, they actually need more resources for them. So many of the countries actually are really suffering with the longevity. So it's not that actually we don't want longevity. When we have longevity, how do we ensure rest of the other things are taken care? So that is one of the main aspects. So when we talk about life, it actually not only we are talking about money, we are also talking about the biological side of the life. So that part actually we'll be discussing in the next few slides after the part of the actuarial science. Now I request Rohan to pick up the actuarial science part of it, and then from there actually we'll move to the main presentation.
Yeah, thank you. Thank you for the introduction as well, sir. So can you please move to the next slide? Yeah, so talking about who are actuaries, right? So generally an actuary, you find actuary having a skill sets of mathematics, statistics, economics, and also some aspects of law, which they apply on their day-to-day basis. And some of the areas where you find actuaries working the most is in the area of insurance. But we would also find actuaries working on various other areas like pensions, right? Like banking, financial sectors, and the very recent, in the very recent times, you would also find actuaries working along with data scientists, more of which I'll be discussing in the later slides, right? So when we talk who an actuary is, so actuaries are skilled in quantifying risks using probabilities, right? So they design solutions to reduce the impact of undesirable events. So to put it in a nutshell, what an actuary helps the organization do is help give some sense of prediction as to what is going to happen in the future. So whichever area requires that skill sets to be used, that's where you find actuaries working the most, right? So which helps them navigate ambiguity in very complex environments in the business setting, helps business make important decisions and strategies, and they are also very trusted advisors to the senior management. Therefore, their skill sets are highly valued in an organization. Can we move to the next slide? So, though you would find actually working majorly in the area of life insurance, health insurance, pensions, but they are just not limited to these areas. So wherever we find the skill sets of risk management, probability, statistics, predicting future events, probability modeling, you would find actuaries working in all of those areas. Hence, we have a list of n number of actuaries on these slides, which is mentioned there. So insurance companies are the primary organizations where actuaries take lead in strategy. And also to highlight, insurance companies cannot function without actuaries. And as I mentioned, right, so in the current generation, we are also finding actuaries working very closely with data scientists, leading data scientist teams also. And you would also find actuaries working in some key analytical roles in some of the top technology companies like Uber, Tesla, Google, Expedia, to name a few. We move to the next slide. So talking about how the entire profession has evolved over various generations, right? So it's not a new profession, though not many people have heard about what actuarial profession is. Let me make it clear, right, that it's not a new profession. It's not something which has just come up very recently. It's just that the awareness has awareness has started very recently, but the actual profession as such, it has been existing since the 17th century, wherein we had the very first kind of actuaries working primarily in the area of life insurance, That's where we had the very first kind of actuaries named life actuaries. Then during the 20th century, when the industrial revolution started coming up, we found the insurance companies found that general insurance is a big market. That's where we saw the second generation of actuaries role being evolved, and that is what is the second kind of actuaries called as the non-life actuary. Then during the late 20th century, where we had people, talking about investments, asset liability management, and derivatives, credits, banking, finance, when all such kind of sectors started picking up, that's where we found third kind of actuaries coming up into the picture, which is where we see actuaries working in asset liability management kind of sector. Then the fourth kind of actuaries which we saw coming up was in the beginning of 21st century, wherein we had actuaries working in the areas of risk, enterprise risk management, so which incorporated, using various kinds of risk-based solvency methods and stuff like that. So that's where we found actuaries, fourth kind of actuaries being evolved. And in the very recent times, over the last 10 decades, we have seen the the profession being evolved, a profession being evolved into the data science sector, wherein we see actuaries working with big data analytics and unstructured data set, working very closely with the data science team. Can you please move to the next slide? So talking about the building blocks of the very recent generation of actuaries, right? The data science actuaries, actual data science, that's what we call it. So it is based on these four fundamental building blocks, which is economics, mathematics and statistics, computer science, and law. Correct? And as I had mentioned, so we find these actuaries working in various kinds of sectors. So to talk a bit more about what actual data science is, when we move to the next slide. So to give you a comparison between how actuaries, actuarial science and data science compares, right? So we find that many of the skill sets of whatever data scientists say is shared commonly between the actual science, but where the actual science professionals shine is in the area of business and domain knowledge. So that's where we find actuaries working more. Nevertheless, we see that currently in almost all the organization, me personally currently I am actually leading a data science team in Accenture, right? So that distinction between the actuarial science and the data science is only becoming narrower and narrower day by day, and the values of the actual professionals are being recognized in the organization. So can you please move to the next slide? So talking about what are the various kinds of skill sets which we see, which currently the organizations are also looking at in the actual professionals. So this is the diagram which very nicely highlights the same. So if you see, the data science can be broadly classified, the various methods in data science that we learn can be it can be very broadly classified into these three segments, which is the infrastructures and tools, concepts and methods, and data. And within each we have various sub-levels. So all the points which is highlighted in green and blue, you can see, right, is the one which is currently are the skill sets which the organizations are currently demanding. Just to name a few, right? We talk about programming languages like R, Shiny, R Markdown, Git, Keras, Python, SQL. These are all the skill sets which the organizations are demanding. To talk about the visualization aspect, right? Like ggplot, understanding of ggplot, Power BI, Tabular, Matplotlib. Then we have also various skill sets that we pick up, like supervised learning based models, unsupervised learning based models. We also upskill ourselves in various computational methods like MapReduce, Hadoop, Scala, to name a few. And then also the data aspects. When it comes to legal ethics and regulations, we need to be aware of what the global data protection regulations are. GDPR is what we call it. Then data security, data ethics, then various storage and modeling techniques like MySQL, and NLPs like text processing, text classification. So these are all the skill sets which the organizations are currently demanding out of the actual profession in their organization. So yeah, I guess that pretty much gives the overview of where the entire the entire profession is currently skilled at, and where the entire profession is currently heading to. And in the coming future, we would find more and more actuaries working in such areas. So with that, if we move to the next slide. So, so we are very proud to tell that this was recognized very early on in our university, and in the year 2017 is when it was first being offered officially to the MSc students as an elective to the Masters of Mathematics course. And since then, the university has produced about 36 research papers and book chapters in the area of actuarial data science, about 27 dissertations in various actuarial streams have been worked on. About 12 or more conferences has been attended or presented in an international forum. About 55 students have passed at least 2 actual exams. 2 PhDs have been awarded in the area of actuarial data science. 4 actuarial data science workshops have been conducted across various forums. 3 tools have been developed for the industry. 2 individual research grant awards have been conferred by the Casualty Actuarial Society and the Society of Actuaries by the US, from the US. 1 student has received an International CAS Scholarship Award, and having recognized all of these accomplishments, last year we were very fortunate to have received a recognition by the CAS in form of the CAS University Award, which was awarded to only top 4 universities in the world. And it's a very proud moment for us to mention that our university was recognized as one. And in fact, our university was the only university in Asia to have got this award. So yeah, I guess it's really proud for us to bring this up. So with that, I would leave it to Sir to take it up from the next slide onwards. Over to you, sir.
Sure. Thank you, Rohan. It was nice, actually, so flowing. So we'll start the main activity of the mathematical biology side, like actuarial science, how it does. So just before starting, we have Bhagavan's message on this area. So he spoke very widely of almost all the required thing. So yeah, so this is related to the body, cells, and coordination, and life and death within the body, right? So please play the audio.
మనకి ఎంత టీచ్ చేస్తుండదో చక్కగా యోచించేయండి. యు ఆల్సో థింక్ ఆఫ్ ద హార్ట్ దట్ గివ్స్ గుడ్ టీచింగ్. కానీ మనం టైర్డ్ అయిపోతున్నాము. బట్ వి ఆర్ టైర్డ్. మనం చాలా విసిగెత్తుతున్నాము. వి ఆర్ డిస్గస్టెడ్. చాలా వీక్ అయిపోతుంటున్నాము. వి బికమ్ వీక్. కానీ దేనివల్ల? వై? నిర్విరామ కృషి చేస్తున్నాము, దానివల్ల మనకు టైర్డ్ అయిపోతున్నాము. బికాజ్ వి డు వర్క్ కంటిన్యూస్లీ, దేర్ఫోర్ వి గెట్ టైర్డ్. ఒరే పిచ్చివాడా, నిర్విరామ కృషియే నీ యొక్క జీవితం యొక్క ప్రధానమైన లక్ష్యం అని హార్ట్ మనకు బోధిస్తుండాలి. ది హార్ట్ టీచర్స్ దిస్ లెసన్. ది కంటిన్యూస్ వర్క్ ఓన్లీ వెరీ ఇంపార్టెంట్ టు యువర్ లైఫ్. దట్ ఇస్ ద లెసన్ టాట్ బై ది హార్ట్. కర్మానుబంధీని మనుష్యలోకే. మనుష్యుడు ఇంత కూడా కర్మతో చేరినటువంటి వాడు. ద హ్యూమన్ సొసైటీ ఇస్ బౌండ్ బై యాక్షన్. శరీర మాధ్యం కలు ధర్మ సాధకం. ద బాడీ ఇస్ గిఫ్టెడ్ ఫర్ రైచియస్ యాక్షన్. కనుకనే నీవు కర్మాచరణ కోసం నీవు జన్మించావు. Therefore you are born for activity. కర్మ చేయవలసినటువంటిది నీ కర్తవ్యము. You should perform your duty. అంతేకాని టైర్డ్ అయిపోయినావని నీవు ఇమాజినేషన్ కు అవకాశం ఇవ్వకూడదు. So you should never imagine that you are tired. అని హార్ట్ మనకు బోధిస్తుండాలి. It is the lesson taught by the heart. ఎట్లా? How? హార్ట్ నిరంతరము కొడుతూనే ఉంటుండాలి, పని చేస్తూ. దీనికి రెస్ట్ లేదు. హార్ట్ బీట్స్ కంటిన్యూస్లీ. ఇట్ హస్ నో రెస్ట్. అయితే దానికి ఒక రెస్ట్ ఉంది. But it too has got a rest to a certain extent. టక్ 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 టక్. ఈ టక్ టక్ రెండు టక్కులకు మధ్యలో ఒక రెస్ట్ ఉంటుండాలి. బిట్వీన్ గెట్ స్పాయిల్డ్. పని చేసింది అది బాగా. ఇట్ వర్క్స్ కంటిన్యూస్లీ. ఈ పని చేసి ఈ బ్లడ్ కొంచెము బ్యాడ్ బ్లడ్ గా మారిపోతుండాలి. Because of continuous work, the blood becomes bad. ఈ బ్యాడ్ బ్లడ్ అక్కడికి పోయి చేరుతుంది. Bad blood reaches heart. అక్కడ ఆ సెల్ ఫుల్ అయ్యేంత వరకు కూడా రెస్టే దానికి.
The cell will be under the rest until it is
అది
full.
ఫుల్ అయిన తక్షణమే తిరిగి ప్రారంభం చేస్తుంది.
The moment it is full, it starts working.
అక్కడ నుంచి పంపు కొట్టి
Then it pumps up.
లంగ్స్ కు పంపిస్తుండాలి.
It sends it to the lungs.
ఈ లంగ్స్ అంతా కూడా అక్కడ ప్యూరిఫై చేస్తుండాలి.
Lungs purify the
ఆ
blood.
ప్యూరిఫై చేసి మరొక వైపున దీనికి పంపిస్తుండాలి.
Then it will send to another side.
ఇవన్నీ కూడాను దానికి రెస్టే.
All this amounts to rest.
ఈ రెస్ట్ లేకుండా పోలేదు.
There is no doubt
నా
rest.
పనిలోనే నాకు రెస్ట్ ఉండాలని చెప్తున్నారు ఈ హార్ట్.
The heart teaches you the lesson that there is a rest while at
అందువల్ల నా
work.
పిల్లలు కూడా నేను చెప్తుంటాను.
That's what Swami tells to students.
స్వామి 4 గంటలకు లేచి రాత్రి 11 గంటలకు మేము చదువుతుంటాము, మాకు రెస్టే లేదే అనుకుంటారు.
They tell Swami, we get up at 4 o'clock and read till 11 o'clock in the night. We have no rest.
రెస్ట్ అంటే ఏమిటి?
What do you mean by rest?
పడక వేసుకొని పండుకోవటమా రెస్ట్?
Is it simply to lie down on the bed?
చేంజ్ ఆఫ్ వర్క్ ఈజ్ రెస్ట్.
Swami says, change of work is rest.
అదే విధంగానే ఇక్కడ చేంజ్ ఆఫ్ వర్క్ జరుగుతుంటుండాలి హార్ట్ లో.
Similarly, in the heart, there is change of work.
ఈ యొక్క సెల్ ఫుల్ అయ్యేంత వరకును రెస్ట్ గా ఉంటుంది. అదైతేనే మరి తిరిగి పంపు కొడుతుంటాది.
The cell will be at rest until it is full. Thereafter, it pumps up.
కనుక ఇంత సూక్ష్మంగా జరిగిపోతుంటుండాలి.
It happens in a subtle
ఈ
way.
బ్లడ్ సెల్స్ ఒక్కొక్క చిన్న సెల్స్ లో కూడా ఎంతో పని జరుగుతుంటుండాలి.
In blood cells, in every minute cell, there is a lot of work going on.
ఎవ్వరు కూడా ఈ యొక్క సృష్టి రహస్యాన్ని గుర్తించుకోలేరు.
No one can understand the mystery of nature.
బాడీ అంతా బ్లడ్ సెల్స్.
The body is full of blood cells.
ఒక్క సెల్ లోపల ఏమిటి ఉండదో అన్ని సెల్స్ లోపల కూడా అదే పవర్ ఉంటుండాలి.
What all that is present in a cell is present in other cells also.
ప్యూరిఫై చేస్తుండాలి.
It purifies.
ఎంత దీన్ని యాక్షన్ చేస్తుండాలి.
It also
తక్షణమే
activates.
ఒకదానికొకటి త్యాగం చేస్తుండాలి. త్యాగం చేస్తుండాలి.
Each cell sacrifices for the other.
ఈ త్యాగం అనేటువంటిది మానవునికి బోధన చేస్తుండాలి.
The lesson of sacrifice is taught to humanity.
ఆ త్యాగం చేసినప్పుడే మనకు క్షేమం చేకూరుతుండాలి.
When we sacrifice, the welfare is ensured.
నా చేయండి ఇక్కడ బ్లడ్ సర్క్యులేషన్ జరుగుతుండాలి.
There is blood circulation there in the hand.
క్షణానికి ఇక్కడ బ్లడ్ దాన్ని వదిలిపెట్టి పోతుండాలి. త్యాగం చేస్తుండాలి.
After a moment, the blood leaves that place. It sacrifices.
అది త్యాగం చేయకుండా బ్లడ్ అక్కడే గిరేపితే బాయిల్ ఏర్పడిపోతుంది.
If it does not sacrifice, the blood clots. You find the boil there.
అది మనకు రోగంగా రూపొందుతుంది.
That becomes the disease.
త్యాగం వల్ల ఎంత క్షేమం జరుగుతుండాలి.
Only out of sacrifice there will be
మన
security.
జీవితం అనేటువంటిది ముందుకు నడచాలనుకుంటే త్యాగం త్యాగం త్యాగం.
If we go ahead in life, the sacrifice is necessary.
న కర్మణ న ప్రజయా ధనేన త్యాగేనైకే అమృతత్వ మానసు.
No action, no society, no position, no wealth would ensure this eternity other than sacrifice.
అమృతత్వ మానసు.
It's only sacrifice.
చేస్తుండాలి.
It takes you to eternity,
Immortality.
immortality.
So, yep, that's for each one to understand the message. There's no need to put any interpretation there. So, one aspect of this sacrifice is actually that happens at this cell. If it doesn't sacrifice to the next cell, it actually transforms into major diseases, including cancer. It works against the system. So, we have 100 trillion cells. That is some range numbers, like some say 40 trillion, some say, this is the number normally we quote for the number of cells in human body. Each of these cells, several of them, they form into tissue, and tissue to organ, organ to system. There are multiple systems we have in our body. So if we have to work on understanding the longevity, we have to work on this cell level. So these cells are of different type in nature. Yeah. So what happens in our body at every moment is actually, the death is not the final last point. Death is actually happening at every moment in our body, because many cells are dying and then new cells are getting produced. So when they start continuing this process, the life remains in the body. When they stop, the rejuvenation stops, then slowly it drops. So this is the concept, that's the turnaround thing which is happening within the body. So this is, you see actually certain cells, they have just 2 to 3 days only. They're okay, fine. So certain cells, they have just 2 to 3 days type of thing only, say 3 to 5 days. So certain cells, they have 120 days. Some of them, they have actually the whole life, like cells in the eye, they have whole life. So when we are calculating the longevity of a person at a given point of time, so we actually need to look at actually how it is the cell part of it is working. And that actually gives a very clear picture of how to arrive at the final number. So when you look at actually the number, roughly 330 billion cells, plus or minus 20 billion, they turn over every day in our body. That means actually these many cells are dying, and then some new cells are coming. So it's a marvel which is happening within our cells on a daily basis. So if we look at that, actually, so we are talking about, so in a couple of months, actually, you are coming back actually into a new person, because most of the things are actually rejuvenated, new ones, the dead cells, and the new ones are coming up. So that's the science which is actually going behind the aging. So unless we get this one, this is there in the modern science, this is there in the Yoga Shastra, this is there in every field of medicine, what happens. So the next stage, what they found is actually how to improve this longevity by taking one organ and putting it one place. So those are the different types of things, then working on the genes, those are the additional things which come into picture. So for us to calculate, have a model, it's absolutely necessary for us to understand how this works. And bones also are considered as actually within that, the bone cells are there, they work in one different way, blood cells work in different way. So each one has their own duty, and they relinquish their thing when the duty is over. If they sustain, then it actually leads into the a disease for us. So that's what actually Swami was mentioning. So if there is no sacrifice from one cell to the other cell, then actually it just becomes a clot, and then it can lead to any other thing. So apart from the accidental and some of the other things which come and shorten our life, we are in a position to calculate in what is the life expectancy using the actuarial science methods. But it is coming from the data. It is not coming from the cell level calculation of it, because there is no model yet to be developed. So 2019, there was an effort actually which was made by actuaries to come up with. So just to give little understanding, when we say 1 crore, it is actually after 7 zeros, that's a big number. When we are talking about 1 billion, it is 100 crores. When we are talking about 1 trillion, it's actually 1 lakh crores. So 1 lakh crores, such of that 100 trillion is what we are talking, our body is composed of. So it's massive even to comprehend what is happening within our body while we are actually surviving. So going forward, so biology of aging, when we say it is accompanied by gradual changes in most systems, so we can classify the respiratory system, blood circulation system, like that. Actually 9 major systems are there, but like there are many more subsystems available in the body. So it is a gradual thing which is happening, and it is ending. Where actually the new cells are not producing, that's where actually the whole thing is ending. But there are certain cells which are actually meant for life. But if we misuse, then they die beforehand. So for getting it to misuse, it's not just the body which is actually impacting us, it's the mind which is actually the boss of the body, which is actually impacting the continuation of the existing cells. So the research is underway in these areas. So basically, most of the diseases, you can actually relate it to which cells are actually not really operating as expected, and then how they are getting impacted with the external stress and other things. So that is the composition which is in the research area now. So now when we look at actually the unresolved problem, this is not a resolved problem. So we are actually discussing this, but it's not a resolved problem. It's an unresolved problem for centuries. There is no model which actually correctly predicts actually what is the aging and quantifying the aging date. Small piece of the body is being identified, something is produced, that's all done. But nobody actually ever solved or came up with a model which actually projects the pattern of aging for our place. But how we calculate, because in the business we do that. So there are like actuarial science, we have life insurance mechanism, or the covering the life risk. So that is where actually we have various models which we use for the mortality and morbidity. Actually we used to be called actual mortality scientists in the few decades back. So we used to work only on the probability of death. And there's a Swami's quotation, if you understand what happens at the death, your understanding is more deeper. There are 3 stages: birth and what all happens, and death. Death is the place where actually everything else closes, and then you'll be able to understand the concept so well. So mortality is one such concept which actually helps actuaries to do. So these are all the things which we do as part of the actuarial science. So mortality table is by age, what is the probability of death? So we have these algebraic formulas, which actually I kept here the deterministic way. These are all actually can be calculated in a random variable method. So where actually we take a radix for a location, out of how many people die of that particular age within 1 year. So that becomes the basis for us to calculate. So every country has its own mortality table. So this is a fundamental requirement for each country to plan their retirement benefits, plan any social security aspects. So this is a most crucial thing. So I left these formulas here, so definitely at some point of time, whenever anyone shows interest, we can actually discuss those things. But this is a mathematical aspect of the mortality table. So here we do graduation, various statistical models we use to get there. So the way it appears is this, and then these are all the graphs we use for various aspects of it. This is for like United States, we have, this is for India, the table is for India. So now the other concept which comes is actually survival curves. Survival curves is actually the way actually Kaplan-Meier thing. So this is the code we use. So we did this entire thing from our end. That's where actually we are showing the snippets of the outcome from us. So this is a very related model for us again. So where actually we will be able to identify time to that particular event. So in this scenario, time to death, that's what actually we are going to calculate. So lot of models are there in this, so which actually help us to identify the appropriate graph. So here actually the actual true trend actually you can find fitted model. So we use data science for our purpose because a lot of data is actually involved in this. So for the entire country we calculate for a given period because the mortality is actually going down. Mortality going down means people are surviving for a longer period. So that is actually a decremental table. So all the calculation they get reversed in the mortality side. So going forward now, another important one is the life expectancy. So you can see life expectancy actually of various countries, which is shown here. And these are the formulas we use for actually calculating the life expectancy. It is the probability of survival multiplied by the given aspect of where it stands in each of the scenarios. So it has 2 types of thing, cut rate and then full. So basically half of the year, if we are taking every moment of life and doing that, that's an integration. If we are doing it at different points, that's actually the discrete way of calculating. We can do both the ways. The difference is half a year for us. That's the approximation we take. So this is a calculation which is again exclusively done by actuaries of their country. So you can see we were actually 45 or 36 actually, 36 to 40 at the time of independence, 1947, and we got to 70. This itself is actually a massive improvement for us. Okay. So moving forward, now using these concepts, this is the place actually where we have type aging model, PTAM we call. So this came up actually in 2019. This research actually published by Society of Actuaries of US, that is one of the world's largest body for actuarial science on the life side. So they produced this particular paper where actually we have used the Markovian model where we actually try to map where it can lead, taking many of the biological aspects into consideration. So it has a unique structure. It includes actually constant transition rate for the aging process. So in the functional form of relationship between aging and death. Okay. So with a shape parameter to capture the biologically deteriorating thing. So if you look at actually the Markovian process, I'll just show 2-3 graphs here. So phase type aging model. So here if you see, so these are the, okay, so I need to be here. So the psi is the one which is actually the T-var, actually this is stochastic model, okay. The tail var calculation at the 0.999 if we do, that age is actually 112.55. So stochastically we actually moved up to 500 years of age. So what happens from the point of view of the model? So this is where actually the red one is actually the true one, and then the remaining ones are actually going at this page, because you can't have life beyond the given point of time. So but like from the stochastic modeling, you can actually produce this type of thing and then see where it actually cuts off for our purpose. So this is another graph actually where you can see the fitted model in the red. So these two, what exactly they indicated is, using this model, the PTAM we call, we are able to get to a realistic one, so which actually works. This whole thing we did for a group of people who are in the retirement, we call actually retirement areas, that is communities, where actually people above age 60, they all stay there for in a comfortable life. So from them actually we calculated these outputs. So this gave us a good confidence. Actually this type of model can work in practice using the actuarial models, concepts which are like the mortality table, because for centuries this particular activity has been done by the actuarial group. So moving them into the biology actually makes more difference. So this is one of the major papers. So 2007 we had one more paper that was on the, like we did the similar thing in the deterministic way. So this one came up actually from the with the stochastic modeling and then recent data science tools and techniques we used, and we came to this. So our paper will explain what all we took and what data, and then what are the parameters we have chosen, and then the limits of that. But here actually we just showed actually the output of it, which actually confirms it works for us. So moving forward, so this particular research actually we want to extend this PTM model for various other age groups, because we have taken the retirement group, and then because that's where actually we have stable population available for us in one particular community, so which actually we can adopt that to different age groups. They apply this one to Indian scenario, where actually we can see how this type of model can be tweaked to see, because the end product of this is actually something we are trying to solve many centuries problem of coming up with a pattern for a mathematical model to see where actually we can, how we can predict the life properly. Because the other way around is what we are doing now. By looking at what happened in the last few years, we come up with a mortality table. But the other way, the correct way to do is go from the cell level and then go to the tissue, then organ. What is failing? That is the model actually, which is not there in the current world. So this is a collaboration. It's actually a lot of interdisciplinary activities are there. So we are actually looking at collaboration within the SSSIHL with other departments on this type of work, and then collaboration with other organizations, because it's of national significance, because identifying a pattern for getting to the life expectancy is of great importance for any nation. So most countries, they work in this area, but different, like economic groups normally work in that, less of mathematical people. So this is where actually we are pitching in to work on this. Then, so these are all the from where actually references, citations you can actually pick up. So the entire information is there. So a lot of calculations are involved. So that's where actually I didn't take you all in this half an hour. So we thought actually first we bring you what is actuarial science, then make you familiar and the type of work which is being done in our organization. So any quick questions from your end, we are ready to take up.
So any questions from the speakers and the participants?
Thank you so much for the nice talk and making us aware about the aging and the responsible factors for aging.
Yes.
So my question is very general. So the first question is, why aging? What is the main reason? Is it due to the stop of regeneration of new cell that you, in the starting you have told? And then how it is connected with the longevity? Actually, what is longevity? So can you just...
Okay. So longevity, the definition, how long people live from their birth in a given place. Okay, from birth to, like at that point of time, if we calculate how many years they are going to live. So that's the, this picture here is from birth. If you calculate expectancy, life expectancy at age 40, life expectancy will be shorter than that, right, depending on that. So that is the definition of life expectancy. So people living longer, okay, that is actually longer than what we normally see is actually, that's what actually we are trying to identify for the purpose of paying a longer time, the pension or any other thing. So with respect to what is the aging, okay, so aging is actually gradual changes in most of the body parts. That is what is actually body systems. And system, when we say each of the, there are like many systems in the body, like respiratory system and other things. Each of these systems actually they start malfunctioning or they stop functioning for various reasons. One of the reasons is actually rejuvenation, okay, that is not the complete rejuvenation of the cell. But there are many other reasons actually why it stops, because the collaboration between one cell, the one system, the other system, that is another reason actually why it stops. Then external reasons like bacteria coming in our way and then stopping that, actually making it malfunctioning, an accident and other things which can come in. Taking out those probabilities, like possibilities of actually external events, if we had put a group of people in a place where actually they are not affected by outside world, the aging is actually the cell has a timeline. That's what actually we said, right? This lives only this much time. So 3 to 5 days, in some cases blood cells and other thing. So when the rate of new cells is not 100%, one cell dies, another cell doesn't take place. It is actually, each part of the body has its own ratio. So based on that, that particular thing will slowly, gradually come down. That's why it's a gradual stopping of that particular thing. So rejuvenation, that's why actually people go for rejuvenation of each part of the body, the yoga and other thing, because those parts of the body are not used for most of the life, so they stop functioning from our end. And the rejuvenation may not be required. That's how body may think. We don't know because it's an unknown world. But the cooperation between the cells and the continuous, like heart cells are there. Like when they stop, the entire body stops. But there are many other parts of the body, when they stop, entire body doesn't stop. So the importance is there for certain cells and not there. Okay, right, sir. I'm fine.
Any more questions?
Yes, sir. Yeah. Japan, my graph actually is shorter, so it cuts actually. It goes beyond 85. So I just cut there. But I know. 85. So they make century. Like, see, when we say these numbers are average numbers of all the people. So 70 means even like most of our known people, they survive up to 85 actually nowadays. So, but there are like those without the proper accommodation and food and other thing, nutritious food. So they may die a little earlier. So it's an average for the country. So our country, if we take actually only elite population, it's actually far superior to most of the other countries also. So yeah, but you are right, sir. So Japan actually tops the list. Most of the people. So our pride is having younger generation in the country. So more younger generation we have. So though population is more, so population is more was considered as a negative thing earlier, but actually it is considered as one of the most positive things in the current world. Now they are realizing having good population, good number of young population, it actually adds value to every country's development.
Yes. Thank you, sir. Any questions from the in-person participants? From the virtual participants, do you have any question? Okay. If not, let us give a big round of applause to sir for the interesting and valuable talk. Thank you, sir. So with this, we come to the end of the technical sessions of the conference. So now I have the pleasant duty of announcing the Best Research Paper Awards. So I request this research, we had around 13 entries for this Best Research Paper Award. So these awards are judged by an eminent jury, being spearheaded by our faculty, Dr. Bharatwaj, and also joined by Dr. Muthu from NIT Warangal and Dr. Varaprasad from VIT Vellore. So I request Bharatwat sir and Muthu sir to come forward to hand over the winner and runner-ups for the Best Research Paper Award. So, yeah, it's kind of overwhelming to see that our own student, Mr. Ritvik Chandra Pandey, secures the winner for the Best Research Paper Award. Mr. Ritvik Chandra Pandey. No, no, give it. Round loud of applause for him. The first runner-up goes to Mr. Vinay Kumar from Shivanadar Institute of Eminence. The second runner-up goes to Ms. Priyanka NC from NIT Tiruchi. Okay. Thank you, sir. Thank you, sir. Thank you all. So we are almost at the close of the conference. So I'm really thankful to many people, of course. Formally, the vote of thanks will be proposed by Mr. Ritvik. So I would like to, we would, in fact, as on behalf of the, when I say I mean on behalf of the organizing committee, I'm speaking. So we would like to elicit any feedback, constructive suggestions from the speakers, the in